Hey everyone, welcome to today's Explainer. We're opening up a fascinating and honestly kind of terrifying deep dive dossier into two highly cunning microbial villains, Leptospira and Listeria. If you're tuning in, you know that understanding pathology isn't just about memorizing a list of symptoms. It's like a giant puzzle. It's about tracking exactly how these microscopic organisms systematically breach, hijack, and dismantle complex biological systems. So we're going to strictly follow our source pathology notes today and break down the exact strategies these two bacteria use to survive in the wild and wreak havoc on their hosts. Okay, let's dive into this structural roadmap. First, we'll look at the unseen animal invaders. Then we're going to tackle Leptospira, the spiral threat, before zooming in on how it impacts dogs and cattle. After that, we pivot to Listeria, the cellular hijacker, and wrap up with the three specific Listeriosis syndromes.
All right, part one, the unseen animal invaders.
Now, you might be wondering where these microscopic battles actually happen. Well, it's not in some isolated, sterile laboratory. We're talking about the environments and animals we interact with every single day. Pastures, kennels, you name it. The core theme for us today is that these are absolutely not just isolated animal issues. We're dealing with two completely distinct pathogens that both use a massive network of animal reservoirs to maintain their existence. And the wild part? Humans are highly susceptible to both. We literally get caught in the crossfire as accidental hosts just by stepping into their transmission cycles.
Moving right along to part two, Leptospira, the spiral threat. So, Leptospira isn't your standard, run-of-the-mill, blob-like bacterium. Our source notes specifically describe them as thin, spiral organisms. But here's the absolute game changer. They're motile by periplasmic flagella. Think of these flagella like built-in internal motors wrapping around the bacteria. It literally lets them corkscrew and burrow their way highly effectively through tissues and fluids to reach their target organs. Pretty devious, right?
Now, what's really interesting about this matrix is the sheer scale of cross-species danger at play. The pathology divides Leptospira into different serotypes, and each one has a specific maintenance host, like rats for Ictereka hemorrhagia or dogs for Canicola. These hosts harbor the bacteria naturally. But I want you to look at the far right column, the accidental hosts. Notice a repeating word? Humans. Yep, us. Whether the original source is a rat, a dog, a pig, or cattle, humans are universally susceptible across all these major serogroups.
Okay, so how does it actually operate once it gets inside a host? Well, here's the clear pathogenesis sequence. Regardless of the species infected, Leptospira has a very distinct modus operandi. It heads straight for the renal system. Step 1, it localizes right in the kidney. Step 2, it multiplies rapidly inside the lumen. Step 3, it gets excreted via the host's urine back into the environment. And finally, step 4, all this intense bacterial colonization induces acute or chronic nephritis, which is a severe inflammation of the kidneys. So you gotta ask, how on earth is this cycle sustained? Get this, when rats are infected with the serovar hemorrhagiae, they don't just get sick, fight it off, and clear the infection. No, they shed the organism in their urine for the rest of their lives. For life. Just think about that for a second. A single infected rat becomes a relentless, permanent environmental dispenser for this pathogen.
All right, let's zoom in a bit more. Part 3, Leptospirosis in dogs and cattle.
In dogs, the pathology actually splits into two very distinct presentations depending on the serogroup. First, we have the acute phase, often called the septicemic stage, which is usually caused by that rat-borne icterohemorrhagic. This one is brutal, and it's fast. We're talking massive dehydration, jaundice, and severe internal tissue destruction where the actual liver cells shrink and completely dissociate, literally breaking up their normal columns. Contrast that with the subacute phase, usually caused by the Canicola serogroup. In this phase, the poor dog typically succumbs to uremia, total kidney failure, driven by intense, significant lesions right at the corticomedullary junction of the kidney. And our source specifically defines this subacute nephritic form as Stuttgart disease. This is an absolutely critical clinical term to anchor in your memory. When you hear Stuttgart disease in canine pathology, you should immediately know we're talking about a subacute Leptospira canicula infection that is aggressively destroying the renal system.
The reality of Stuttgart disease is, frankly, just devastating. The pathology notes provide this very vivid sensory detail for the post-mortem presentation. An animal that succumbs to this is going to show severe dehydration and emaciation.
but most notably, a very strong uremic smell. Because the kidneys have totally shut down, all those toxins that should have been filtered out have just built up systemically.
Now, the pathology looks a bit different, but is equally devastating in cattle. Bovine leptospirosis creates focal lesions in the kidney parenchyma, basically a chronic nephritis that results in what pathologists call a white-spotted kidney. It's a highly visual marker. And the devastation absolutely doesn't stop there. In pregnant cows, this infection often causes abortion in the latter half of pregnancy. Meanwhile, in young calves, the acute disease can trigger severe hemolytic anemia.
Okay, time for a sharp pivot. Section 4. Listeria, the cellular hijacker. Meet Listeria monocytogenes. Now contrast this directly with Leptospira. Where Leptospira was a thin spiral, Listeria is a rod-shaped, gram-positive bacterium. But its most defining and honestly most dangerous characteristic is its battleground. It is an intracellular organism. It doesn't just swim around outside our cells. Its entire survival strategy relies on forcing its way inside them.
Let's move on and see how this builds into one of the most terrifying cellular prison breaks in pathology. Check this out. Step 1. The bacteria have surface proteins called internalins that act like a master key, binding perfectly to host E-cadherin receptors on epithelial cells. Step 2. Once locked on, they penetrate, multiply, and destroy the cell. Step 3. When the host's immune macrophages rush in and eat the bacteria, Listeria springs its trap. Step 4. It literally escapes the macrophage's internal phagosome prison and survives and multiplies inside the very immune cell meant to kill it. And Step 5. It can now travel directly from cell to cell without ever being exposed to extracellular fluid or our circulating antibodies. It uses our own immune system as a Trojan horse. Crazy, right?
Because of this incredibly stealthy cell-to-cell movement, Listeria can execute a shocking path of infection. When animals, especially ruminants, get tiny mucosal injuries in their mouth just from nibbling tough feed, Listeria gets in. It then utilizes centripetal movement within the branches of the trigeminal nerve to physically climb the facial nerve pathways, straight up into the brain, specifically targeting the medulla oblongata.
Which brings us to our final part. Section 5. The 3 Listeriosis syndromes.
The first and perhaps most famous manifestation is circling disease. This is the clinical definition of the encephalitis syndrome, mostly seen in ruminants when the bacteria actually reach the central nervous system. Because the brainstem itself is infected, the poor animal will exhibit an abnormal, twisted posture of the head and neck and will just compulsively, aimlessly walk in circles. So, to summarize this precisely, here are the 3 distinct syndromes driven by Listeria. First up, the encephalitis we just discussed, striking the central nervous system of ruminants, but also horses, dogs, pigs, and yes, humans too. Second is systemic infection, or septicemia. This generalized infection is most common in monogastric animals and human newborns, and it's characterized by focal necrosis of the liver. And third, abortion. In pregnant cattle and sheep, particularly in that crucial last quarter of gestation, the organism penetrates the placenta, reaches the fetal liver, multiplies, and unfortunately causes fetal death.
Well, that brings us to the end of today's explainer. We've mapped how Leptospira corkscrews its way through kidneys, and how Listeria hijacks our very immune cells to climb right into the brain. But I think the absolutely crucial takeaway here is looking at the sheer interconnectedness of it all. As we map out the complex pathways of Leptospira and Listeria, we really have to ask ourselves, living alongside these massive animal reservoirs, from the rats in city alleys to the cattle out in the fields, just how fragile is the barrier between their diseases and our own? Thank you so much for focusing in with us today, and keep questioning the unseen world around you.